These are the lecture notes for Chapter 37 on neurons and the nervous systems. And again, as with the other body systems, we are going to focus on the human nervous system. You can see some other animals and the way that the nerves are highlighted in yellow, so you can see the way they are arranged. The hydra, which is a cnidarian related to a jellyfish, has a nerve net. And then the planarian, which is a flatworm, has a ladder-like nervous system with a collection of nerves in the head region called cerebral ganglia. The earthworm has a true brain and a ventral nerve cord rather than a dorsal nerve cord like we have. And then you have the crab, which has a brain and then also thoracic ganglion, which looks to be even larger than the brain, honestly. But that's just a collection of nerves and nerve fibers in the chest region of the crab. And then we have the squid that has a brain. And then, of course, a cat's going to be similar to most other mammals. It has a larger brain and containing a cerebrum like we do and a spinal cord. So for the human nervous system, it's divided into the central nervous system and the peripheral nervous system. The CNS includes the brain and the spinal cord, and the PNS is everything else. Mostly what we're talking about there are like the cranial nerves and the spinal nerves. There are two divisions of the PNS. The somatic nervous system controls activities and controls skeletal muscle. So it is involved in anything that affects skeletal muscle movements. That is somatic. So somatic is skeletal muscle. Autonomic nervous system is all the other types of muscle. So all of our organs and pathways like our digestive tract and our airways and our blood vessels that are lined with smooth muscle, and also our cardiac muscle for our heart, and then glands. All of these are controlled by the autonomic nervous system. And the autonomic nervous system, or ANS, is divided into the sympathetic and parasympathetic divisions. So if we look at this diagram here, let me make it bigger. You can see the central nervous system is the brain and the spinal cord, and then the PNS is divided into the somatic and the autonomic. So the somatic nervous system deals with skeletal muscle and anything that affects skeletal muscle contraction. And the autonomic nervous system deals with cardiac muscle, smooth muscle, and glands. And again, it is divided into the sympathetic and parasympathetic divisions. One thing that distinguishes primates from other mammals is that the frontal lobe, or the forehead, the region that's correlates to the forehead, is large and complex. And primates have increased brain capacity. There's increased brain capacity have allowed mammals, especially humans and other primates, to become adept at higher mental activities. So it's that frontal lobe that separates the primates from the other mammals. Now, nervous tissue is divided into neurons, which are nerve cells, and neuroglia, which are the support cells for the neurons. A neuron cell body is like any other cell body. It contains a nucleus and organelles like mitochondria. The dendrites extend out from the cell body. There are many, many branches, and they extend out from the cell body and receive signals from sensory receptors or other neurons and take those signals to the cell body. So dendrites are receiving signals and carrying those signals to the cell body. And the axon conducts a nerve impulse away from the cell body. Typically is covered by a myelin sheath, which is like insulation, and it allows for faster conduction. And any long axon is referred to as a nerve fiber. The neuroglia or neuroglial cells, let's start with astrocytes. The astrocytes help maintain the health of the neurons, and they maintain what we call the blood-brain barrier. They keep the blood from contacting the actual surface of the neuron, because the blood carries chemicals and pathogens, bacteria, all kinds of things that could destroy a neuron. And when our neurons are destroyed, we don't get them back because they don't divide. Then we have microglia, which are phagocytic cells. In other words, they engulf and destroy harmful substances like bacteria. So they're like part of the immune system. In the central nervous system, we have cells that make the myelin sheath that surrounds the axon, and they are called oligodendrocytes. That word again is oligodendrocytes. And in the peripheral nervous system, the cells that perform that same function are called Schwann cells. So both of these, oligodendrocytes and Schwann cells, form the myelin sheath around axons. The gaps between the myelin sheaths are called nodes of Ranvier. And then we have ependymal cells and satellite cells. The ependymal cells produce the cerebrospinal fluid, and the satellite cells surround neuron cell bodies in ganglia, which means these are found in the peripheral nervous system. They kind of have a similar role as astrocytes, but for the PNS, whereas astrocytes maintain the blood-brain barrier and do the same thing for the spinal cord in the central nervous system. So satellite cells and astrocytes are very similar, but the satellite cells are part of the PNS and the astrocytes are part of the CNS. And satellite cells do participate in responses to injury. Now, there are different types of neurons. The motor neurons or efferent neurons are the ones that transmit a signal from the central nervous system to muscles or glands. And they're always multipolar neurons, which means they have many dendrites in one long axon. Then your sensory neurons or afferent neurons receive impulses from sensory receptors and take those impulses to the central nervous system. And they are unipolar neurons. Interneurons are only found in the central nervous system, the CNS. They are multipolar, and they form complex pathways in the brain and enable us to think, have memory, and language. Now, this is the anatomy of a multipolar neuron, many dendrites in a single axon surrounded by myelin. And then this is a unipolar neuron. This is a sensory neuron, which is unipolar. You can see the cell body is separated from the extensions. So you have two axons that extend away from the cell body, and one of them functions like a dendrite, and the other one transmits the signal to a target. So here you can see a typical neuron, the cell body, the dendrites, the axon. Now, to understand how a nerve impulse works, a nerve impulse is an electrical signal. We also call it an action potential. So to understand action potential, you first have to understand resting membrane potential. The resting potential is the voltage across the plasma membrane of the neuron when the axon is not conducting an impulse. In other words, when it's at rest. And for the resting membrane potential, the inside of the neuron is more negative than the outside, and the voltage is around negative 70 millivolts. And the sodium-potassium pump plays a very important role in the resting potential. It helps to maintain the resting potential by pumping sodium ions out and potassium ions in to the neuron. So the resting potential is negative 70 millivolts. That's the potential when, that's the voltage across the axon membrane when the axon is at rest. But an action potential is a change in membrane potential across the axon, across the plasma membrane of the axon. We call it the axonal membrane. And that change causes the negative 70 millivolts to go in a more positive direction all the way up to positive 30 millivolts. So an action potential is generated only after a stimulus larger than the threshold. So the threshold is normally a stimulus that has to raise the voltage or potential from negative 70 to somewhere around negative 55 or negative 60 or negative 55. So once that threshold is reached, the action potential will be generated. The components of the action potential, in other words, the events that occur during an action potential, are depolarization, repolarization. So the depolarization part is when the negative voltage changes to a positive. It says the inside of the neuron becomes positive because of the sudden entrance of sodium ions. And this reversal of polarity, in other words, the reversal from negative to positive, causes sodium channels to close and potassium channels to open. When potassium channels open, since potassium is, the level of potassium inside the neuron is high, potassium is going to leave. And that's going to lead to repolarization. When potassium channels open and potassium leaves the axon, the membrane potential returns back to the resting potential of it that has a negative value. So we can look at these pictures, and this is just a portion of the axon at rest. You can see that it's got negative charges built up on the inside of the axonal membrane and positive charges on the outside. And if we connect a voltmeter, the voltage across that axonal membrane is negative 70 millivolts. You can see even bigger, you can see the orange, the little orange ions are going to be sodium, and then the pink ions are potassium. So you can see inside the axon has a high concentration of potassium due to the sodium-potassium pump. It's constantly pumping sodium out and potassium in. And then the outside has a higher concentration of sodium. So what happens if we open sodium gates that allow sodium to cross the membrane? It's going to go from where its concentration is high to where its concentration is low. So once those gates are triggered and they open up, normally by a neurotransmitter binding to them, then sodium is going to enter, and it's going to cause that voltage to become more positive. The sodium ions are positive, so it's going to cause that resting membrane potential to go toward negative 55, which is the threshold. Once negative 55 has been reached, many sodium gates open. So sodium rushes into the cell, and that causes the voltage or potential to increase to positive 35. And at that point, the sodium gates will close or inactivate, and the potassium gates will open. And potassium concentration is high inside, so potassium is going to leak out. And since potassium also has a positive charge, when you lose those positively charged potassium, when they leave the axon, losing positive charges means your voltage is going to become more negative. So the potassium is going to leave the neuron or the axon until the voltage is back to negative 70. That's repolarization. So depolarization is moving, the voltage moving in a more positive direction, and repolarization is when it goes back into a negative direction. Depolarization happens because sodium comes in to the axon, and repolarization happens because potassium moves out of the axon. Now the next section is on the propagation or the spread of the action potential throughout the entire axon. So if an axon has myelin, then the conduction is called saltatory conduction, and it's much, much faster. And if it doesn't, if the axon is non-myelinated, then it will be continuous conduction, which is slower. But conduction of a nerve impulse is an all or nothing event. And action potential either happens or it doesn't happen, but it always goes to positive 30. Like there's not like a stronger action potential. But the signal, the intensity of the signal is determined by how many impulses are generated within a given time span. So more impulses are going to lead to a more intense signal. There's also a refractory period, which is a time when another action potential cannot occur. And this is an image of the myelin surrounding an axon. And you can see there are areas that are not myelinated, and those are called nodes of Ranvier. So the action potential literally jumps from node to node to node, and that's what enables it to go so much faster. And we're going to stop here for this first part, and